we're developing the immune therapies. A lot of immune therapy historically has been antibody-based, and the problem with an antibody is they can be very, very effective. They're very specific, but there's a half-life. I think what everyone's been excited about over the last decade is the ability to capture and utilize living cells, the immune cells, where you can actually genetically modify an immune cell, infuse them back in, and where you'd be able to get a therapeutic benefit. But in this case, the cell is a viable cell that can be sustained in the body, so the half-life potentially is not just days or even weeks, but there have been some studies now showing that years later you can find a genetically modified immune cell that's still functional in the person's body. So in this case, they've identified a series of diseases that have very, very low likelihood of any meaningful survival. And also they have to have a common antigen. It doesn't make sense if you have a very specific cancer antigen to that specific cancer. That becomes a specific product for that patient. What has been the focus of attention is, can you identify a specific molecule commonly shown across all cancers that could then be a target for immune destruction? And I think that's the basis of chimeric antigen receptor T-cell therapy. They'll take your own lymphocytes, genetically modify it by inserting a gene that will express a new receptor that will redirect that immune effector cell to the actual cancer itself to destroy the cancer. Yeah, so the methodology of the study is it's identified as a patient population which is of large cell lymphoma, 75,000 cases of lymphoma a year, 1/3 of them are large cell lymphoma. 60% will be cured potentially by chemotherapy and antibody therapy. 10,000 patients a year in the United States, will go on to need further therapy, and a lot of them will die of their disease. This particular study was focused as a group of patients that have multiply relapsed or have been proven to be resistant to primary therapy. And in this case, these patients we would have predicted is probably in the average about 15%, maybe 20% 2-year survival. And the ability of getting to complete remission again in this group with the standard chemotherapy and antibody therapy is less than 10%. And it was felt that recognizing it's a high death rate, it's a common disease, and that there's a common target antigen that we can address and develop a redirect the immune system toward, that this may be an opportunity to change the natural history of that disease. And I think that's our study. There's been other similar studies using similar constructs and similar molecules, but I will point out that if you look across the landscape of chimeric antigen receptor T cells, they are different drugs. Ours is using the molecule tisagenlecleucel, or the cell product tisagenlecleucel, which is and we ran a clinical trial sponsored by Novartis that came out of the work from the University of Pennsylvania. And the data show that you can achieve a much higher response rate than that would have been anticipated in this group, and a higher complete remission rate that would have been otherwise expected. I think it's exciting in that if our data, like others, are now showing a 1.5 years out from the administration that half the patients are remaining free of progression, when you would have predicted, based on any other prior chemotherapy or antibody-based therapy, that there would have been much more early relapses and most of the patients would have died by that time. So in our study, of the patients that were infused, 40% of patients were able to gain a complete remission, 14% had a partial remission, so a total response rate of 54%. I think what was intriguing, and it goes again to the biology of the cells, over half the patients with partial remission actually proceeded to a complete remission. So this shows that you can actually get a biologic activity over time. And the survival rate, if you take the entire group, approximates between 35% and 40%. But for the patients who had a remission status, there's over 50% survival ongoing currently as of now. The study continues to accrue. I believe the data that we presented was based on approximately 100 patients. Any therapy will have its own adverse events. There are some unique adverse events of interest, and this is one. The first is what's called cytokine release syndrome, and that's an inflammatory event that occurs when the T cell encounters its target. The inflammatory event will be associated with sometimes as extreme hemodynamic instability, or sometimes it can be very minor, low-grade fevers, a little heart rates that go faster. Sometimes people have had difficulty oxygenation and required intensive care unit support. In general, the cytokine release syndrome was able to be managed. I think it's also unique in our study is approximately 20 to 25% of the patients were able to be treated in an outpatient setting without a significant toxicity of CRS. There's also a lot of attention that there's a relatively still relatively not understood well toxicity of a neurologic toxicity of delirium and encephalopathy. Again, that was seen, but in about 10% of patients. And again, I think one of the when you look at cross studies, it's sometimes hard to draw good comparisons because there's different grading systems have been used, and I think we're moving as a community to a consensus grading system. So what we report today will be different than what we report tomorrow. But I think that so the cytokine release, the inflammatory system, and encephalopathy, and then there are some patients who have a prolonged what we call cytopenia. There's a bone marrow toxicity that we typically predict after chemotherapy that marrow will recover function within, days or maybe 1 or 2 weeks. And there are a small number of patients that have a profound, persistent low blood counts, often requiring transfusion and setting them up for infection. Well, I think the implications are, in some ways obvious. You have a therapy that has changed the natural history of disease. When we would have predicted, 15 to 20% survival at 2 years, now we're predicting approximately 40% survival at 2 years. I think the implications are, it's a first step. At the end of the day, that means, to me, 50 to 60% of patients still aren't surviving. And what we need to do is to figure ways to build upon this platform. It's a first step, it's not a final step.